The vaccine your dog receives, the parasite treatment that protects his heart, even the insulin that could one day keep him alive, none of them began as ordinary medicine. Each started at a moment when doctors or veterinarians faced a disease they could not stop. These are 4 medical discoveries your dog might one day owe his life to. And the final one came at a cost few owners know. It is July 1885. 9-year-old Joseph Meister has been bitten more than a dozen times by a dog believed to be rabid. Once symptoms appeared, rabies was considered almost invariably fatal. There was no proven treatment that could save him. His mother brought him to Louis Pasteur, a scientist who had spent years developing an experimental vaccine through research involving infected rabbits. The treatment had worked in animals. It had never been given to a human. Pasteur was not a physician and he could not legally treat the boy himself. A doctor named Jacques-Joseph Granchet administered the injections under Pasteur's direction. Over 10 days, Meister received a series of preparations made from infected rabbit spinal cords, beginning with material in which the virus had been weakened the most, then progressing carefully toward stronger preparations. It was an extraordinary risk, but rabies offered almost no alternative. Meister survived. That survival did not instantly create the modern vaccine your dog receives today. The method, manufacturing, and safety of rabies vaccines would continue to evolve for generations. But the principle had been demonstrated. The immune system could be prepared before the virus reached the brain and caused irreversible disease. Today, canine rabies vaccines safely expose the immune system to rabies antigens, allowing it to recognize and fight the real virus later. What feels like a routine injection at the vet rests on a discovery made when preventing rabies in a human being still sounded almost impossible. And the stakes extend beyond the dog receiving the shot. Vaccinating dogs is one of the most effective ways to interrupt rabies transmission to people. One protected dog can become part of a barrier protecting an entire community. Almost a century later, the emergency was no longer one child. It was an entire population of dogs. By 1978, veterinarians across several continents were confronting a newly recognized disease spreading with terrifying speed. Puppies were hit especially hard. A dog could begin with vomiting and diarrhea, lose fluid rapidly, and become critically ill within a remarkably short time. Researchers were still trying to understand what they were seeing. Cornell's Baker Institute was flooded with calls, letters, and biological samples from veterinarians searching for answers. The culprit was identified as a new canine parvovirus, closely related to viruses already known in other carnivores. It targeted rapidly dividing cells, especially those in the intestinal crypts, bone marrow, and lymphoid tissues. That combination damaged the intestinal barrier while weakening the body's ability to replace immune cells. In a young puppy, the result could be devastating. The speed of the scientific response was extraordinary. Researchers identified the virus, developed diagnostic tools, studied how it spread, and moved rapidly toward vaccination. Early vaccines were followed by improved formulations as scientists learned more about the new disease and the protection dogs needed. Today, parvovirus vaccination feels like one of the most ordinary parts of puppy care. In 1978, veterinarians were confronting a disease new enough that they were still learning its name. Vaccination does not make every case impossible, and a sick puppy still needs urgent veterinary care. But the reason modern owners can protect dogs before exposure is that researchers and veterinarians responded to a global canine emergency while it was unfolding in front of them. The third discovery traveled in the opposite direction, from parasite research in animals to one of the largest treatment programs in human history. For much of veterinary history, heartworm infection was difficult to prevent. A dog could appear healthy while worms developed inside the pulmonary arteries, gradually damaging the vessels and forcing the heart and lungs to work harder. By the time obvious signs appeared, treatment could be more complicated and the disease more advanced. The breakthrough began with soil. Japanese microbiologist Satoshi Omura collected soil samples and screened the microorganisms inside them for useful compounds. One sample, taken near a golf course not far from Tokyo, contained a bacterium unlike anything his team had previously studied. Samples were sent to William Campbell and his colleagues, who found that compounds produced by the bacterium were extraordinarily effective against parasites. Those compounds became known as avermectins. A modified form, ivermectin, would become an important veterinary anti-parasitic, including at carefully controlled doses in some heartworm preventives. But the molecule's story did not stop with animals. River blindness is caused by a parasitic worm transmitted through the bites of blackflies that breed near fast-flowing rivers. Repeated infection can cause severe itching, skin disease, visual impairment, and permanent blindness. Ivermectin provided public health programs with a treatment that could be distributed on an enormous scale. In 1987, the company behind the medicine committed to donating it for as long as necessary, wherever it was needed, for river blindness. The Mectizan donation program continues today and became one of the largest and longest-running medicine donation programs in history. In 2015, the discoveries behind the avermectins were recognized with the Nobel Prize in Physiology or Medicine for William Campbell and Satoshi Omura. A molecule that may protect a dog from parasites also became one of humanity's most important tools against a disease that had damaged lives across entire communities. A soil sample, a bacterium, a veterinary medicine, then a global public health campaign, all connected by the same discovery. The final breakthrough is different. It went on to save millions of human lives, but dogs paid part of the price for discovering it. In the summer of 1921 at the University of Toronto, Frederick Banting and Charles Best worked with experimental dogs whose pancreases had been surgically removed, inducing severe diabetes so pancreatic extracts could be tested. The work was difficult, imprecise, and by modern standards painful to hear about. The extracts were crude. The dogs became gravely ill. Some died before the researchers had anything effective to offer them. On July 27th, one injection produced a clear drop in blood sugar. It was not yet a treatment, but it was evidence that something inside the pancreas could control glucose. Later, a dog remembered as Marjorie survived for roughly 70 days after pancreatectomy, with repeated injections of pancreatic extract. Her survival showed that the effect could be sustained, not simply observed once. The importance of these experiments does not erase what the animals endured. Understanding the history means holding both truths at once. Dogs suffered during the research, and the discovery that emerged would later save lives on an extraordinary scale. The next challenge was making the extract safe enough for a human being. Biochemist James Collip joined the team and developed a cleaner purification method. In January 1922, 14-year-old Leonard Thompson received the first pancreatic extract. The initial preparation produced little benefit and caused an adverse reaction. Collip continued refining the process. On January 23rd, a more purified preparation successfully lowered Thompson's blood sugar where the first attempt had failed. The transformation was profound. Before insulin, a child diagnosed with type 1 diabetes could be placed on a starvation diet that might prolong life but could not stop the disease. Insulin turned a rapidly fatal diagnosis into a condition that could be treated. Experiments involving dogs helped make that breakthrough possible, and today, when a diabetic dog receives insulin from a veterinarian, the discovery returns to the species that helped prove it could work. Not as a perfect circle and not as a story that erases the suffering involved, but as a reminder that the histories of human and veterinary medicine have never been completely separate. Rabies vaccination, protection against parvovirus, a molecule that transformed parasite treatment, insulin. Today, these can feel like ordinary medicine, but each began at a moment when doctors or veterinarians had no reliable answer, and each changed what became possible for the dogs who came afterward. Which of these 4 stories surprised you most? Tell me in the comments, and subscribe to Woof IQ, because behind many of the treatments we now take for granted is a story most dog owners have never been told.